When we look at carbohydrates, the importance that they serve in a biological function is that they are the fuel and building block materials for the cell itself. They include sugars and what we call polymers of sugars. That's a chemistry term that you should know. A polymer is when you take a bunch of very similar units, add them together to make a bigger unit. We have different classifications of carbohydrates, the simplest being monosaccharides, also known as simple sugars. We have disaccharides that consist of two of the smaller units, the monosaccharides. And then we also end up having the polysaccharides, which are going to be these large molecules themselves. Carbohydrates are organic, once again, they're composed of carbon, and they always have this ratio of CHO in that order. It's for every one carbon, there's two N hydrogen and then N oxygen. And that's approximate. It doesn't have to be perfect. So, for example, you could have something that's simple as C6H12O6, or maybe it's something like C6H H12 hydrogen 24 oxygen 11. You can basically see that 1 to 2 to 1 ratio. And like I said, it usually is an approximate. It doesn't have to be perfect. Okay? And once again, they typically all that formula. You can see some examples. The monosaccharides that you need to know. We have glucose, fructose, galactose. There's another one that I'm going to add to that you should know. Ribose, because of its important. I have it on the next slide. And then deoxyribose. Okay, you'll see those in the next slide. The disaccharides, they're composed of double sugars, and they typically have 2 or more monosaccharides. It is important for our test that you're able to figure out or know what the disaccharides are, but also identify them, knowing what they're composed of, what they're made of. So knowing that sucrose, also known as table sugar, is composed of a glucose and a fructose, maltose, glucose and a glucose, lactose, a glucose and a galactose. These are the examples of the monosaccharides that compose the disaccharides. Polysaccharides are sugars that have a many single sugars bonded together. Some common ones that we need to know: starch, glucose, cellulose, chitin's another one. The one that's common in us is that glycogen is stored in the liver, and cellulose is what actually makes the cell wall, and we actually can't digest it. And that's why you can't just eat a grass, for example. A good example of cellulose is in celery, the thin strings of celery that you're unable to digest. That is an example of cellulose. Here's the major monosaccharides that you should be able to know and identify based on how they appear and how they look. So I gave these in our notes: glucose, fructose, galactose, ribose, and deoxyribose. Notice glucose here, galactose are almost identical to each other. They look very similar. They're isomers of each other. They have the same chemical formula. Their structural formula is different, and what's different is this region right there. Seems like a small change, but it creates a huge difference for us. We also have fructose, okay, and then we also have our deoxy and ribose sugars. Those are important to look at. They are similar to there. Notice the subunits at certain locations are different. The biggest difference between deoxy and ribose is that the deoxyribose has been deoxygenated at this location right there. Here's just more images of them so you can kind of actually see them on the test. Don't be surprised in the short answer section if I give you an image and I cover up the particular name and you got to tell me what it is. So you'd have to look at this and say, hey, that's fructose based on its structure. There's glucose there. Now notice glucose can also be a chain in that case, or it can be a ring molecule as well. Excuse me, this is galactose, and then glucose is up top. All of these are these simple sugars, and this is what's actually going to enter into the cell, be broken down through cellular respiration, and get us our ATP, our energy source for us. Here's the 3 big ones. Notice they're highlighted the difference between them, between the fructose, galactose, and glucose. The double bonding. Notice that in this case, that the double bond is actually occurring within the ring, within the chain, excuse me. There's that double bond. It's an easy way to identify fructose versus the glucose, galactose. Notice those are difficult here to look at the differences between those. That's something that you'll just have to kind of memorize for the test. I will be willing to bet, though, I'm not going to put both these on there. You know, I'd assume that's an easy one to tell the difference. You should be able to identify that. There's another example of the monosaccharides again, the fructose, galactose, and glucose. You should have those. What I want you to do in your notes, you should be taking notes as we're going through here. So if you haven't, make sure you get a piece of paper out. You'll bring this in. That'll be part of your homework. So go ahead and pause that and get that paper out. What I want you to do now is to pause, and you can go back if you need to, but I want you to write out and draw the structure for the major classifications of the monosaccharides. Your fructose, your glucose, your galactose, your ribose, and your deoxyribose. So that's one thing I want you to do now. Pause it and draw those. For the DNA, deoxyribose, you'll have to go back in the slides or pull it off the internet as well. So go ahead and pause that now. Here's just another image of the different ones that you can see. These are going to be our disaccharides, multiple sugars. So, for example, here, in the case of maltose, to make a maltose, it's two sugars together. Notice we have a glucose on one side, a glucose on the other. It's bonded. In the process of bonding those together, we're going to lose a hydrogen, and oxygen, more commonly known as H2O, and that gives us maltose plus that water. We call this type of reaction a dehydration synthesis. Dehydrate, you lose water. Synthesis, take two smaller pieces, piece A, piece B, and you synthesize or build a bigger molecule. Okay? So that's an example of our first disaccharide, maltose. You can see it there, its actual structure. Two glucoses put together, commonly found in a lot of breads and carbohydrates, what we typically think of as breads and carbohydrates. Two polysaccharides that are important are going to be, you can see here, amylose, amylopectin. Notice these are all glucose. All those little rings in there, they're all glucose molecules. They come together to make one super large molecule that we call a polysaccharide. There's an image of cellulose that can be found in a plant. That's the string, as I said, that's commonly found in the walls of celery, and therefore, so those are the examples of the disaccharides that you need to know. You'll need to know for the test, the sucrose is composed of glucose and fructose. Maltose is composed of two glucoses. Lactose is a glucose and a galactose. Key thing with here, by the way, if you haven't noticed, all carbohydrates end in ose. So most of them, their names will end in ose. That tells you it's a sugar-based molecule. The polysaccharides have specialized names. The ones that you'll need to know for the test, just what they are. You got your starch, your cellulose, and your glycogen. That's a big summary there. Make sure you put in the notes after you drew those monosaccharides, put this information right there. You don't have to draw them. You should have them already drawn. So put the sucrose, what it's composed of, and then just put the names of the three types of polysaccharides that you need to know. The last thing you need to know about carbohydrates, and it's not in your notes, so make sure you write this down in your homework that you have and know this, is that the bond that connects two molecules, so I got a glucose here and a glucose here. Remember, I'm going to combine those together in the process and lose water in a dehydration synthesis. This bond right here, what we call that is a glycosidic bond, the glycosidic bond. So once again, it's a bond that connects two glucoses or two other simple sugars. That is the glycosidic bond. And if you have to remember this, we'll talk about it a little bit later. The type of reaction that's occurring that bonds these together is called a dehydration, lose the water, synthesis reaction. Our next type of organic molecule we call a lipid. Okay, lipids are considered fats and oils. The other classification that we have is waxes. And they are also composed, just like before, carbon, hydrogen, and oxygen. So make sure you have that down in your notes. So there's CH and O. The difference is this right here. Carbohydrates, they end up having a 1 to 2 to 1 ratio, whereas lipids do not have that 1 to 2 to 1 ratio. I like to call it more of a 1 to 2 to approximately none or 0, right? Approximately. There's not none, okay? There's not 0. There's just very little. The example that I give you, C57, H110, oxygen 6. Much less than the other two. That's what we're referring to. If you're produced by an animal, we call that lipid it's a solid fat. Plants are usually liquid. So a good example, lard is an example of a fat. When you take lard and you have it after you cook, let's say you cook meat on the stove, you'll notice that if you don't wash that pan, that fat that was a liquid starts to solidify or become solid. The plant ones are liquid, so like canola oil, olive oil, those are liquids. Those are good examples. Waxes can be produced by both. Now, let me clarify this. It doesn't mean that only animals produce solid fats and only plants produce liquids, but overall, the major fats produced by animals tend to be solids. The major fats produced by plants tend to be liquids. So it's a generality. It's not a all the time scenario. Those are some examples of some lipids. Fats, right? They're solid. A couple big things that you need to know with fats are this information. I'd write this down and bring it to class. Is that they're composed of 3 fatty acids and 1 glycerol. They have a carboxyl group, which is this down right here. Now, just as we talked about before, there's special types of bonding that are associated here. So what ends up happening, why this is important, is that type of bond. So we have the fatty acid portion and the glycerol. The glycerol typically is there's 3 of them. It kind of looks like this. There's a carbon here, a carbon here, a carbon here. There's an oxygen, there's an oxygen, there's an oxygen. We got your hydrogens. That's going to represent like that, those hydrogen little line. And then what we have is a carbon bonded here to another oxygen. And then we have these chains of carbon. This is the fatty acid portion. Okay? Let me move that over and shrink it a little bit. So it's long chains. And I'm not going to do this for all of them. It's the same concept like that. And a lot of times we represent them just like lines like that. Now what's important to note is if all of these bonds in this region, not this double bond, we're not talking about this double bond here or here, but in this region that I circled, if they're all single bonds, it has become fully hydrogenated with hydrogen, so there's full hydrogen. We call that a saturated fat, okay? So what I just drew down here is a saturated fat. If at any place in this region down here, there's 1 double bond or more, so if I were to put a double bond right there, there's no longer an hydrogen fully saturated with hydrogen, and therefore we would call this type of fat a unsaturated fat, an unsaturated fat. Now, you may have heard those terms, unsaturated and saturated fat, and they mean a lot of different things for us in biology. And that's what we're going to talk about for the last little bit of slide. In this case here, what do we have? I want you, and I'm not going to tell you, I want you to tell me, is this a saturated or unsaturated fat? So in your notes, just put down below what we have already, is this a saturated or unsaturated? I'll give you a second to think about it, and just as a tip or a hint, remember you are going to look in this region for something. You're going to look for a double bond. And whether there's a double bond in this region here, not these double bonds, that doesn't count, but in this region here, it'll tell you whether it's a saturated or unsaturated. Take a look here. Notice there's no double bonds. Here we have 1 double bond, and the book doesn't represent it, but if I were to make this slide, I would put maybe a little line right there. Why do I know there's a double bond right there? It created a kink in the structure. So whether you actually see a double bond like that, or you just see the kink action, that tells you it's a double bond. That makes this a unsaturated. And when we think about unsaturated, there's characteristics that unsaturated fats have versus saturated. Here it is, unsaturated fats. Okay? unsaturated fats. Unsaturated fats come from unsaturated comes from plants. Okay, saturated, that's the bad ones, right? They tend to have high heart and circulatory diseases. And so a couple things that you need to know about the saturated versus unsaturated that you should write down. They come typically from plants. They're oils, which make them liquid at room temperature, right? They're healthier for us. Sorry, I got bad. It's right on my finger. It's hard. They're healthier. Those are some characteristics you should think about. The saturated fats, on the other hand, tend to come from animals, right? They tend to be solid at room temperature. Solid. And they tend to be unhealthy for us. Unhealthier, I guess would be the way to say it. Okay? So that's what we're referring to when we're talking about saturated versus unsaturated fats. Cholesterol is also an example of a fat, and we'll talk more about that in class. That's where I want to stop the video. The rest of these slides we'll pick up with time, and you can kind of take a look at saturated versus. This is the slide that we'll kind of end with today. Thanks for watching. Make sure you bring in the notes that you took over this video that I kind of told you to record as you go. Any additional notes are fine to turn in as well. Saturated